Welcome to the deep dive. Today, we are plunging into, well, the very forces that govern your life. We're going to be jumping from the explosive minute-by-minute chaos of physical mechanics straight into the quiet ancient language of our fundamental genetic code. Our mission is to look at sources that show how two fields that seem miles apart, biomechanical simulation and molecular genetics, are both trying to tackle immense biological complexity. And they're doing it by, in a way, seeking a kind of sophisticated simplicity.
It's a real study in extremes, you're right. We're going to start with a massive clinical problem, which is blood clotting. I mean, if you look at hypercoagulable states or thrombophilia, as it's sometimes called, these are conditions where your blood just clots way too easily.
And for you listening, that is a clear and present danger. We are talking about really serious life-threatening events, things like deep vein thrombosis, DVT, where clots form in the deep veins, and then the devastating risk of a pulmonary embolus, stroke, or a heart attack if one of those clots breaks free.
And these disorders can be inherited. We see conditions like factor V Leiden or the prothrombin gene mutation running in families, or they can be acquired from things like cancer, trauma, even certain medications. The coagulation system is your body's safety net against bleeding out, but if it runs too hot, it becomes deadly.
Right. So the system itself is incredibly complex, and it's incredibly fast. Let's start there in section 1, modeling the coagulation cascade. If we want to study this, say, to predict a patient's risk or model how a clot forms in some winding, diseased artery, like an atherosclerosis, we have to be able to simulate it.
And that's exactly where computational biology just runs straight into a brick wall. The human system is dynamic, it's huge. Traditional computational models, they try to use what are called convection reaction diffusion equations to map out the whole process. You're typically looking at 30 to 40 different chemical constituents, proteins, mostly all reacting, all moving around. Sometimes it's hundreds.
Okay, 40 variables sounds bad enough on its own. What makes tracking them so, I guess, computationally impossible?
It's the scale. It really is the scale. These proteins exist at concentrations that vary wildly. I'm talking orders of magnitude. You go from picomoles, which is incredibly rare,
Almost nothing.
all the way up to nanomoles.
Yeah.
And trying to solve for a real protein that's reacting instantly, while also solving for the much slower, larger scale movement of blood flow. It creates what we call stiffness issues in the math.
Stiffness issues, that sounds like a very technical term for this is basically impossible.
Essentially, yes. Imagine trying to time a hummingbird's wing beat, right, which is milliseconds. But at the same time, you're trying to track the movement of a glacier, which takes years. If you run both simulations at the tiny time step you need for the hummingbird, well, the glacier simulation would take centuries to finish. That huge difference in speed and scale makes simulating it in 3D in realistic conditions totally infeasible.
So the goal isn't to create a perfect digital twin of a person, but a tool that's actually usable, one that's predictive. And this brings us to the core insight of the research you shared with me, the absolute necessity of simplification.
Precisely. Researchers need a simplified model so that their predictions can actually be tested in a lab. And critically, so they can run simulations of the really complex physical mechanics, like how a clot forms inside a real artery.
Okay, so let's unpack that. This is the crucial simplification. The source material talks about a shift from tracking dozens of proteins down to focusing on only 4 key variables, all related to thrombin. That feels like a huge leap.
It is aggressive, but it's functional. They focus on the master enzyme of clotting, which is thrombin, and its immediate regulators. So you have thrombin itself, prothrombin, which is its inactive precursor, antithrombin, the main inhibitor.
Right.
And then the complex they form when they bind together. The thinking was, if you can accurately model the key player, maybe the supporting cast isn't as critical for the prediction.
But wait, reducing, 40 variables down to 4? Aren't you just throwing out too much important information? How can that even work?
That's the beautiful part. They didn't just throw out data. They built their knowledge of the system's phases into the model. They created a switching model. They knew the process has a very slow initiation phase, which is then followed by a much, much faster propagation and termination phase.
And how does that switch work in the model? What's the trigger?
The trigger is a concentration threshold for thrombin. As soon as the thrombin concentration hits 2 nanomoles, the model basically flips a switch. It changes its internal math to match the rapid propagation phase. It's like a relay race. The slow runner hands the baton to the sprinter at exactly that 2 nanomole mark. It lets the simulation run orders of magnitude faster without losing that essential behavior.
So the simplification didn't just make the math easier, you're saying it actually revealed a hidden behavior that the more complicated model might have actually obscured.
Exactly. And here's the real validation of the whole approach. This simple model robustly predicted something called sustained thrombin activity. Think of it like a process that's supposed to shut down completely, but instead it just keeps idling, kind of dangerously. When you have this specific chemical imbalance, too much prothrombin compared to antithrombin, the termination phase never fully stops. The clotting activity just persists at a low, non-zero level.
And this prediction of persistent, dangerous idling, this sustained activity, that's been confirmed in experiments, right?
It has. And this is a critical tipping point. The researchers think the sustained activity is paramount when you're studying clotting in, say, compromised flow conditions, like stasis, where blood is pooling and you get less of that antithrombin inhibitor flowing in. That persistent thrombin, even if it's low, could easily spark an unwanted clot downstream. It just shows that smart simplification can find the real essence of the chaos.
That is fascinating. So you simplify the physics to understand the hidden chemistry. But the chemistry itself, all those proteins, thrombin, antithrombin, they're all governed by an ultimate biological blueprint. And that brings us to the code itself.
Exactly. We're moving from predicting the protein's mechanical behavior to understanding its actual instruction set. The whole process depends on proteins, and proteins are generated from instructions encoded in our DNA.
And if we're going to talk about biological blueprints, we have to start with the origin of genetics. Gregor Mendel. He's often seen as this historical figure, but his work was profoundly mathematical.
He really was the originator of the idea that traits are discrete, quantifiable things. Back in the mid-19th century, he was doing these systematic experiments with pea plants, tracking 7 different traits.
Yeah.
Tall versus short, round versus wrinkled seeds.
And what he figured out, the very concept of a gene, it changed everything.
Absolutely. He inferred that characters are controlled by 2 factors, what we now call genes or alleles. One would be dominant, the other recessive. And critically, he established that these factors separate when gametes are formed. His quantitative observation that the recessive trait pops back up in the second generation in a roughly 3-to-1 ratio, that was the mathematical key that unlocks the whole logic of inheritance.
So Mendel cracked the logic, but the actual physical stuff that carries that logic, the famous double helix, that wasn't revealed for almost 100 years.
That's right, 1953. James Watson and Francis Crick presented the double helical model of DNA, building heavily on the X-ray diffraction images from Rosalind Franklin. And that structure is where all these genetic instructions for things like clotting factors actually live.
We all learn about the twisting ladder in school, but the real magic isn't just the twist, it's the specificity of the rungs inside.
That specificity is the source of all of life's fidelity. DNA is made of nucleotides, a sugar, a phosphate, and one of 4 nitrogen bases, adenine, thymine, guanine, cytosine, and the rule is absolute. A only pairs with T, G only pairs with C. That pairing ensures that when the helix unwinds to make a copy, it has this built-in, self-checking, error-minimizing system. It's the core reason the code is so reliable.
And that code is the definitive instruction set for every single protein in your body, from the enzymes that digest your food to the proteins that fight infection.
Exactly. Genes are just segments of DNA that contain the recipe for a protein. The instruction set is carried out by RNA. You have messenger RNA, or mRNA, that carries the info out of the nucleus to the cell's protein factory, the ribosome, and then transfer RNA, tRNA, brings the amino acids, the building blocks, to assemble the final protein.
And if we look at our own defense systems, this precision is what allows us to create these incredibly specialized defense proteins like antibodies.
Antibodies are a phenomenal example of this, or immunoglobulins. They're produced by B lymphocytes, and they're these Y-shaped proteins with incredibly specific binding sites. They're designed to recognize and neutralize one exact antigen or toxin. The sheer sophistication needed to create millions of unique antibodies, each for a different threat, all relies on the precision of that DNA blueprint.
And the moment we understood that blueprint, the conversation really shifted from just reading the code to rewriting it. This is where we move into the, frankly, breathtaking and complex world of genetic engineering.
Well, biotechnology itself is ancient. We've used microorganisms, yeast, for making bread since 4000 BC. But genetic engineering is totally different. It is intentional, targeted control over traits by making specific changes to an organism's genetic makeup.
So if we want to manipulate that code, we need tools, molecular tools. What are the key enzymes that let us perform surgery at the DNA level?
You basically need a system for cutting, pasting, and transporting. The molecular scissors are restriction endonucleases. These are enzymes that recognize a specific sequence of bases and cut the DNA right there. The glue is an enzyme called ligase, which joins the cut pieces back together. And since you can't just shove DNA into a cell, you need a delivery vehicle, a vector. Plasmids, which are these tiny circles of DNA in bacteria, are the most common vectors we use.
And this capability has just revolutionized medicine, especially in making therapeutic proteins.
Absolutely. I mean, think about proteins that are too complex or too rare to just extract from nature. We can now take the gene for human insulin, put it into a bacteria, and turn that bacteria into a tiny factory that just pumps out insulin for diabetic patients. And this technology extends to making interferons for viral diseases, somatotropin for growth disorders, even endorphins for pain.
And maybe the most profound application is gene therapy, trying to fix the problem right at the source.
That's the ultimate hope, isn't it? To remove a defective gene that's causing a disease and insert a functional normal copy. It holds immense promise for curing genetic diseases that right now we can only manage. And this research is also expanding into creating what some people call farm animals.
Farm animals, as in the animals themselves become pharmaceutical factories.
Exactly. Researchers can insert a human gene, say, for growth hormone or insulin, into livestock like cows or pigs. And then those therapeutic human proteins can be produced in and harvested from the animal's milk or blood. It's a very complex but potentially more scalable way to do it.
And if we're talking about rewriting the blueprint, we have to talk about the absolute cutting edge. These gene editing systems that have this incredible precision.
We have to be talking about CRISPR-Cas9. This system is revolutionary. It uses a guide RNA molecule to find the exact spot in the genome that needs to be edited, and then the Cas9 enzyme acts as the precise cutter. This is basically molecular word processing. You're not just cutting and pasting big chunks, you're changing individual letters in the code.
The potential is just staggering. We've already seen news reports about children whose genes were edited for specific disease resistance, which, opens up these immense scientific avenues, but it also raises global alarms.
It absolutely does. The technical scope is almost endless, but the societal response is fractured, to say the least. Our sources highlight that worldwide there are organized protests, there's significant scrutiny. The concerns range from potential for exploitation to worries about dangerous long-term effects and, deeper philosophical arguments about whether we should be violating the inherent nature of a living thing. The power is undeniable, but so is the responsibility.
On a different, more applied note, the same fundamental principles that our genetic code is unique allow us to identify individuals with certainty.
Right. That's DNA fingerprinting, or profiling, which was pioneered by Alec Jeffreys way back in 1984. The arrangement of our nucleotides, specifically in these non-coding repetitive parts, differs enough between people to create a unique molecular signature. It's just like a physical fingerprint. And it's now a cornerstone of forensics, paternity disputes, identifying victims after disasters.
So we began this deep dive seeking simplicity in this biomechanical chaos, reducing a huge 40-variable system for blood clotting down to 4 key players and a single switch. And we've ended up exploring the almost infinite precision that's required to manipulate the very DNA code that dictates those 4 players to begin with.
The whole journey, it just confirms that biology is governed by these complex nonlinear dynamics. But underlying all of that complexity is the fundamental precise code of genetics. The real challenge is bridging those two worlds, the world of big-picture prediction and the world of microscopic precision.
When the Human Genome Project finished its mapping, it revealed that the human genome has about 24,000 functional genes. But here's the number that I just can't get out of my head. There is only a tiny, tiny 0.2% difference in DNA among all humans on Earth.
It's an astonishing fact, isn't it? A fraction of a single percent dictates all the variation we see across our entire species.
So if we connect that back to our discussion of CRISPR-Cas9, this enzyme system that can manipulate genetic code at the most fundamental level, we have to ask this: what are the full long-term implications of manipulating that incredibly small 0.2% difference? Not just for treating known diseases, but for defining the future limits and capabilities of our entire species and the balance of life on this planet. That is the question that sits right at the intersection of modeling chaos and rewriting the code.